Before we actually get into the topics of behavioral ecology this semester, I wanted to quickly talk about the three basic methodologies that you can use to test hypotheses uh, uh, in behavioral ecology. Now, this is going to be a review for some of you. For the rest of you, this is important in two ways. One, you should have a better appreciation for the topics that we're going to cover throughout this semester because we're going to be talking about a bunch of case studies, real studies, and how did they actually get their data. And then also for everybody, hopefully this will stimulate some ideas for how to collect and analyze data since you're coming up with your projects right now. So hopefully this will help. Okay, so let's talk about behavioral ecology as a science. With just like any field in science, what you have to do is formulate testable hypotheses. That's a very important step, and you have to use your base knowledge to come up with those hypotheses, which really are educated guesses. I know some people don't like using that term, but I think it is a good description. Then what you do is you find some way to collect data that then you test your hypothesis to see if your hypothesis is supported or if you can reject your hypothesis. And in behavioral ecology, we have access to three broad classes of methods to test hypotheses. The first is oftentimes referred to as observational methods or natural experiments. And what you're doing here is you're looking at natural variation in some trait of interest and looking at the consequences of that variation. Second method is using experimental methodologies. So whatever trait of interest you have, you manipulate that one trait and try to leave everything else the same, and you look at the consequences of that experiment. And then the third method is sometimes referred to as a phylogenetic or comparative method. And in comparative methods, what we're looking for are patterns of evolution, patterns of trends in evolution, or patterns of convergent evolution. So one of the key tools to use in comparative methods are phylogenetic trees. All right. So to kind of demonstrate each of these classes of methods, I want to use one example. And let's look at one behavioral phenomenon, try to come up with hypotheses associated with how it exists, why it exists, and then ways that we can test our adaptive hypotheses. So moths in the genus Automeris display eye spots on their hind wings when they're startled. So in the picture here, you can see the photograph on the left. The forewings are covering the hind wings, and if this moth was on a background, say, of fall leaves, it might very well be pretty well camouflaged. However, under certain stimuli, it will expose those hind wings, which have these bright eye spots on them. And so we have to try to understand this behavior. So the first set of questions we can ask is, how does this behavior occur? So again, we're talking about proximate mechanisms. We can form hypotheses of the proximate mechanisms. For example, expression of gene X influences spot morphology and gene Y affects wing movement patterns. Okay, that would be a hypothesis explaining how this behavior is produced. Another proximate mechanism hypothesis would be initiation of the display is triggered by, and then you'd fill in with whatever you think is the trigger. The proximate trigger to this, is it when the moth is touched by something, so it's a tactile thing? Do they see the approach maybe of a predator and that elicits the behavior? Is it a scent? But you're putting it in the context of, this is kind of a combination proximate ultimate. You're trying to understand what starts the display, but you're thinking it has something to do with maybe avoiding potential predators. That leads us into the why question. And remember, when you're talking about asking why questions, you're really asking or forming hypotheses that deal with the ultimate adaptive function of the trait in question. So, in this case, again, a logical hypothesis would be that the sudden display of these eye spots on the hind wings startles a predator, which increases the chance that the moth that has this behavior will have increased survival and reproduction. That means more fitness. Okay, so how do we test hypotheses? And we're going to really, again, because in this class I'm going to focus primarily on that second set of questions, more the why questions. We're going to kind of restrict our situation to that. So let's first look at doing just a natural experiment or using the observational method, just using the natural variation that exists to try to understand the function of the behavior. So you can ask the question, do individuals differ in their survival abilities due to variation in the eye spot display? So do bigger eye spots affect survivorship so that those with the bigger eye spots create a greater startle effect in the predator? Is it more important to have more contrasting colors so that those with more contrasting colors versus those with less contrasting colors have a better survival? Wing flick rates. If they open up and display the hind wings quick, is that a greater startle effect? Or does that really not matter? Just as long as you get there. So these are not relying on any kind of experimental manipulation of the organism. You just have to have enough variation in the individuals in the population to be able to look for patterns. Now, let's say that you didn't just have any variation. Maybe all of them basically are really fast at exposing their eye spots, that they're all basically the same size. I mean, if there's just not any natural variation, you can't use the observational method. And that's one of the things that people oftentimes debate, which is the best method. Well, really, there isn't a set method that's better than the others. It's really dependent on the circumstances. If there's plenty of variation in nature and it's easier to make observations of those individuals in nature, that might be the route to go. But if we are stuck with a situation where there's just not very much variation out there, that's possibly a sign that there's been very strong selection on that trait in the past, and it's already weeded out all but the best trait variants that exist today. And so in that situation, if it's possible to do experimental manipulations, that might be your best guess or your best bet. So let's kind of see how we might do that with the moths here. Well, one of the things that you could do is you could take moths, and one group of them you just leave them unmanipulated, right? And you do experiments where you put them in the context of certain avian predators, and you see what the response is, and you kind of see what the survival rate of those individuals is. And then you take a second group of moths that you control for their size, age. You try to keep everything consistent except the eye spot. And in this case, what you're going to do is you're going to paint over the eye spot, okay, and try to make it match the rest of the background color of the hind wing. But you're just getting rid of the eye spot. Again, then you expose these individuals to predators, and you see what happens. And so these colored over ones might look something like this. And again, if your hypothesis is that the eye spot confers some advantage in scaring off a predator, you would expect the mortality rate of the individuals that you painted over the eye spot should increase, right? And you may get great results and find out that the individuals that you painted over, they never survive. And you think, this is going to be an excellent publication. I'm going to send this right in. Well, that's not going to be publishable. And I'll tell you the reason. If you ever do an experimental manipulation, it's crucial that you have an experimental control. What the experimental control is it's another group where you use the same methods that you used in the experimental manipulation, but you try to get the individuals in this group to look or behave just like the individuals that are unmanipulated. Okay, now let's try to explain why that is. What the experimental control does is it tests for the potential that your manipulation itself is giving you the results that you're getting. So in this case, perhaps the paint that you use to paint over the eye spots makes the wings stick together. Okay, and in that case, that may in itself be the reason why these moths can't escape the predators. Okay, it doesn't have anything to do with the presence of eye spots or not. It's just the fact that this paint's sticky. Or perhaps the scent, the solvents that are in this paint, make the moths loopy or increases the attack of the predator. Okay, I mean, all those are possibilities. Now, if the thing is, though, if you use the same paints on the experimental control group as you did on the group where you painted over the eye spots, you're controlling for that, right? If the experimental control equals or has very similar results to the unmanipulated group, then you're good to go. Then your experiment is a success, and you can chalk up the result differences you have with the increased predation associated with the painted over group that you took the eye spot away. You can say that's because the eye spot's gone, not because of the experimental manipulation itself. If, however, you find out that the experimental control has more similar results to the group where you painted over the eye spot, well, that's telling you there's just problems with the experimental methodologies themselves, and you really need to go back to the drawing board. Okay, the last thing I want to talk about is using comparative methods. And again, in comparative methods, what you're doing is you're looking for patterns of evolution among multiple lineages. You're looking for increases or decreases through evolutionary time, or patterns of convergent evolution in the same ecological context. So in this case, we want to know, does the behavior, in this case, using wing spots as a potential way of deterring predators, is it used in the same ecological context? In this case, the same may be predator class. So I've got a little made-up example here where we have different species of moths, and you can see some of them do have eye spots. So there's eye spots in 1, 2, 3, 4 species. But others don't have eye spots, and then others have some other traits that we'll talk a little bit more about later. Well, if we look at the predators of each species, we see that in these 5 species, we have warblers as the primary predators. And how warblers forage is they're what we call gleaners. They kind of hop around trees and bushes looking for a stationary prey that they then grab. Okay. And what we see here is that out of the 4 species that have eye spots, all of them have warbler predators. Okay. Now, there's not always a one-to-one correspondence here, and it may be the case that sometimes you have eye spot less individuals, ones without eye spots, that are also being depredated by warblers. Okay, but you're looking for kind of the general pattern. What do the majority of the data reveal to you? Now, if you look at the other species represented up here, you see that the ones with kind of the bright reddish colors and patterns are typically attacked by flycatchers. Flycatchers are not gleaners. They wait for things to fly away, and then they catch them midair in what we call sallies. All right. Well, now that we have this distribution of predators among the different species, and we know how the species are related to each other, we can map on the likely pattern of evolution of different predator classes. So if we're going to use these little tick marks, these little bars, to indicate where we see the evolution of flycatcher predators and warbler predators, the most parsimonious explanation we can have for the distribution of these predator origins is like this. Okay, the easiest, the simplest, most parsimonious is what we call it, explanation for where warbler predators first started attacking this clade, or the representatives in this clade, is that it's a pretty old trait. That the ancestral lineage of these moths probably had to deal with warblers as predators. Okay. Subsequent to this evolution, this one subclade that is primarily with these red butterflies, really their ancestor had to deal more with flycatcher predators, with one exception, this one here, where we have the evolution again of an association between this moth species and a warbler. All right, well, what does this tell us? Well, what it is saying is there is a significant association between those with the eye spots and having warblers as predators. And it's put into an evolutionary context. And what I mean by that is we can't use each species as an independent data point. For example, these two species here, you can't count them as separate because they're so close related. It's more likely that the reason they have eye spots is because they come from a common ancestor. So it wouldn't really be fair for the analyses for you to count these twice. So one of the things about comparative tests is it just kind of helps with your statistical rigor to make sure that you're making appropriate independent comparisons, and that's a key assumption in statistics. But this also makes a very nice story. I mean, it's basically saying that warbler predators evolved were very early problem for these moths, and one of the solutions from the majority of the species was to evolve the eye flicks, the eye spot flicks. And that makes sense, right? If these are a gleaner, you're sitting there, your first camouflage, you have camouflage defense. If that doesn't work, then you do the eye flick to try to scare the predator away while you can escape. Okay. Now, that basically disappeared in most of the species that then were facing flycatcher predators. In the case of a flycatcher, if you're flying around, crypsis is already gone. Camouflage isn't going to really do you any good. And so another tactic that we're going to see that some animals use to avoid predation is to basically do the opposite. Advertise that you're there, but that you're unprofitable. So these red moths are demonstrating what is called aposematic coloration, or warning coloration. They're basically saying, hey, you don't want to eat me because if you eat me, I'm going to make you sick, or I'm killing you, I'm dangerous or in some regard. And in butterflies and moths, when you see this type of pattern, it's usually an indication that they eat plants that have some bitter agent that is distasteful to the potential predator or toxic to the potential predator. But what's really interesting is the one exception to this rule in this clade is this one species that has eye spots that is also attacked primarily by warblers. So see, you're just looking for consistencies in the story to see every time you have an evolutionary change in a predator, do you get the expected change in the moth's response to that? So if you're talking about an evolutionary arms race between predator and prey, do you see a consistent story? That's what the comparative method is doing. It's looking at how similar the stories are in this coevolutionary arms race between independent lineages. Okay, let's review. So we've talked about the fact that behavioral ecology is a science, and so in science you have to come up with falsifiable hypotheses that you then must test. But that means you have to gather data. So what are the methods for collecting data? One we talked about was observational methods, and they work well if there is natural variation in the trait that you're looking at in a natural population, in which case you can just observe that variation and look at the consequences of individuals with different trait variants. There's no need for an experimental control group because you're not doing any manipulations. You get to collect the data in a natural context. But one of the main problems is you may not be able to control all associated variables, and so you have to really think about the timing and how you collect variable your data, like with weather changes, time of day changes. You can't always control those things. Another method we talked about was experimental methods. In this case, there's no need for natural variation in individuals because you're actually manipulating the traits of interest and looking at the effects of those variants on the individuals. You do need an experimental control because whatever method you're using to cause these variations, you need to make sure that those aren't causing some abnormal behaviors in the organisms, and if they are, then you're going to see that your experimental control group is too different from your unmanipulated group. The good thing about experimental methods is it does allow you to conduct the experiments in a controlled environment where you can basically take charge of all the background variables and keep them similar. But in some ways, people will question whether experimental methods sometimes are too artificial. If you're taking the animal out of its natural environment, is it not behaving correctly because of that? So that may be something to consider. The last set of methods we talked about are called comparative methods. This is where you're testing adaptive hypotheses in a historical framework. The main tool you need for this is a phylogeny, so that allows you to document patterns of trait evolution and check for independent lineages of comparison. Sometimes people refer to this as controlling for phylogeny to make sure that your statistics are valid, that you're looking at independent comparisons. One of the biggest problems with using comparative methods is the fact that phylogenies for all groups are just not available right now. So you may be limited in your ability to use this method.